Welcome to The Explainer. Today we're diving straight into the foundational tools of modern biotechnology. Now, you probably already know the basics, but we're going to synthesize the core mechanics here, the ones that are literally transforming our world, from the medicine in your cabinet right down to the food on your table. So, let's get into it.
Okay, let's dive into this. Here's a quick roadmap of where we're headed. We'll start with the biotech revolution, then look at the two pillars of biotech, the precision advantage, that massive 1972 breakthrough, the three-step process, and finally, we'll wrap up with endless possibilities.
Section 1. The Biotech Revolution. Integrating technology with biological systems. So, biotechnology is this truly revolutionary integration, right? It's fusing cutting-edge tech directly with living biological systems. Think of it kind of like a master builder's tool belt. To create something completely novel, you need the tools to modify the underlying architectural blueprint of an organism. Plus, you need a massive physical factory to actually build and multiply that product. These two mechanisms, well, they have to work in perfect tandem.
Moving on to Section 2. Two pillars of biotech. The core approaches driving the field.
All right, so our very first pillar here is genetic engineering. Basically, this is the precise editing of an organism's biological blueprint. By modifying DNA and RNA, scientists can introduce altered genetic information directly into a host organism. We aren't just crossing our fingers and hoping for the best anymore. We're literally selecting and manipulating specific genetic attributes to achieve a highly targeted phenotype.
But, the blueprint is really only half the equation, and that brings us to Pillar 2, bioprocess engineering. Once you've got this genetically engineered microbe, you got to scale it. Bioprocess engineering is what maintains the highly controlled, ultra-sterile environments required during production. It's essentially an advanced microscopic manufacturing plant, a sterile clean room that prevents contamination and allows those specific cells to thrive and multiply on an industrial scale.
Because without this massive scale, modern biotech kind of falls flat. Bioprocess engineering is absolutely crucial. It facilitates the mass production of the bioproducts we rely on every single day. We're talking about the antibiotics curing infections, the vaccines protecting entire populations, and the critical enzymes used in industrial manufacturing. You absolutely need both the genetic blueprint and the sterile factory to make the whole shebang a reality. Section 3. The precision advantage. Why targeted editing matters.
And this brilliantly illustrates exactly why genetic engineering was such a massive leap forward. Because before it, we relied on the clunky, genetic gamble of traditional breeding. Mixing whole organisms meant you were always risking the introduction of highly undesirable traits right alongside the ones you actually wanted. But genetic engineering, it bypasses that gamble entirely. We can now isolate and insert only the beneficial, targeted genes, entirely avoiding all that unwanted genetic baggage.
Which leads us to Section 4, the 1972 breakthrough, a milestone in recombinant DNA.
Now, to really understand how this incredible precision became possible, we have to rewind to a major historical turning point. The year was 1972. This was the year researchers Stanley Cohen and Herbert Boyer successfully developed the very first artificial recombinant DNA molecule.
Their groundbreaking experiment was actually remarkably elegant. They took a desirable gene, in this case specifically for antibiotic resistance, and successfully attached it to a circular piece of DNA called a plasmid. By doing this, they proved that, hey, we could actively combine genetic material from entirely different sources to create something completely functional and new. There was no way we could do that before.
But how did they physically cut and paste microscopic DNA? Well, they used restriction enzymes, which are perfectly described as molecular scissors. And they're brilliant because they don't just chop randomly. No, they cut strictly at highly specific targeted sites, which allowed for the seamless combination of that resistance gene directly with the plasmid DNA. So, once you actually have this new genetic blueprint, how do you deliver it? That's where that circular plasmid acts as a vector. Think of it kind of like a mosquito. Much like a mosquito acts as a delivery system transmitting a disease to a host, the plasmid vector serves as a microscopic vehicle. It carries our newly engineered genetic material straight into the target organism.
Let's move to Section 5 and see how this builds. The three-step process, from identification to maintenance.
In practice, this entire manipulation process boils down to a really clear three-step roadmap. Step 1, you identify the specific DNA harboring your desirable genes. Step 2, you introduce this new DNA directly into the host organism using tools like our trusty plasmid vector. And then step 3, you maintain the introduced DNA within the host. This ensures it can successfully replicate and pass those traits on to future generations.
Let's look at a tangible example of this in action with E. coli. When scientists introduce recombinant DNA into a bacterium like E. coli, the alien DNA integrates and replicates perfectly alongside the host's own genetic material. And thanks to their rapid multiplication rate, this enables the massive, large-scale cloning of those highly beneficial genes.
Finally, Section 6, Endless Possibilities, Unlocking the Future Across Sectors.
Here is the ultimate takeaway. When you combine the pinpoint molecular precision of genetic engineering with the massive, sterile scale of bioprocess engineering, the real-world impact is absolutely staggering. We're now equipped to manipulate vital genetic resources to develop incredibly valuable bioproducts. I mean, this synthesis is driving unprecedented agricultural resilience and extraordinary medical breakthroughs that are literally saving lives right now.
And that brings us to the end of our explainer. The principles of genetic engineering and bioprocess engineering truly underpin the vast potential of modern biotechnology. We now have the blueprints, the molecular scissors, and the biological factories. So I'll leave you with this thought. With these foundational tools firmly in our hands, what do you think will be the next revolutionary trade we introduce to shape our future? Thanks so much for joining me today.